Hey, welcome, Eric. Today we're speaking with Eric Hoffman, somebody I've known for a very long time, feels like almost all my life. I knew Eric when he was at the University of Pittsburgh, and then at one point in time, after many years at Pittsburgh with so many people and his colleagues that were working on Duchenne, he moved to Washington, D.C. And then I think together we understood the value of advocacy. So Eric, welcome.
Thank you, Pat. It's a pleasure to speak with you. It has been quite a long, productive run. I really enjoyed working with you and Parent Project.
Thank you. So the MD Care Act is 20 years old. December 18th, Bush signed it into law. And I remember when you moved to Washington and we first sat down and talked about what would a piece of legislation, if you could get it passed, what would it look like? And you talked about centers of excellence, and we talked about standards of care, and we talked about a plan. And that became the Muscular Dystrophy Care Act. So you were one of the first centers, Wellstone Centers. Eric, can you talk about that first center, what it did and what it was designed to do?
Sure. Our Wellstone Center was a lot about building infrastructure. At the time, as you know, Pat, early on, there just wasn't much going on with Duchenne therapeutics. And part of that was people almost didn't know where to start. And one of the key things is to understand the disease so that you know when you're helping or show efficacy with a drug trial. And so part of that is doing natural history studies. And so both our Wellstone and the Department of Defense, as well as Parent Project and others, funded the largest natural history study to date that was initiated all the way back then. So that was with the Cooperative International Neuromuscular Research Group, or Synergy, a sort of self-formed group of about 40 sites in about 15 countries that all the academic centers had a strong interest in Duchenne, but the Wellstone allowed us to really bring everybody together internationally and start a 400-patient natural history study so that we could get all those milestones and outcomes and lab results so that in the future, when a drug trial was done, we had something to compare the drug effect to and to see if a drug worked and showed efficacy. So the Wellstone was critical in getting that Duchenne Natural History Study up and off the ground. Craig McDonald led that for many years, and many physicians, families, and also foundations participate in that. And now FDA uses that data set as a benchmark to, again, see if drugs are robustly working in DMD patients because they can compare the drug effects to the natural history effects. So that was one of the things. The other thing we initiated through the Wellstone was the early vamorolone studies. So trying to understand how corticosteroids work. They do work in Duchenne, but they come with a lot of side effects. If we understand how they work, can you maybe make a better corticosteroid, one that has the same or better efficacy, but hopefully improve safety outcomes? So that was also the start of the long march of about 10, 12 years to develop vamorolone, which is now, we've recently finished our double-blind registration trial, and that looks to be heading towards drug approval and hopefully will improve patient lives through less toxic side effects seen with many of the corticosteroids. So there's quite a few things that our Wellstone really helped myself, Terry Partridge, Kanabina Nagaraju. We also started cores. A core is like a centralized laboratory with a lot of expertise that can serve not only the investigators working in the Wellstone locally, but then nationally and even globally. And some of that was biomarkers. So can you get blood from Duchenne kids and look at the serum and see different biomarkers that may show progression or severity or how the muscles feeling or doing through a drop of blood without taking a biopsy? And so again, that leveraged many studies with Yetri Hathout and others on Yuan Chen, all on understanding and using biomarkers to facilitate clinical trials. So that also was really initiated by the Wellstone and continued for many years afterwards.
Yeah, it's pretty incredible with Synergy's natural history study. I think it was the first time, first of all, any young man in this case was in a system of continuous monitoring, right? To understand what that person's progression looked like in relation to others. And certainly having that right now as a natural history to benchmark against all clinical trials is pretty impressive. I think that even having 40 sites and outside the and inside and outside the US working together was probably the first time that ever was envisioned. So that is pretty spectacular. Also, the biomarkers that you mentioned, I think that they did a study not only across the trajectory, but now they're focusing in on certain subsets like little guys and see what those proteins are and how they differentiate. So do you think that there's a protein signature or some way by looking at blood proteins, you could understand a person's trajectory a little bit better or predict it?
Yeah, the biomarker work in Duchenne has really expanded. Quite a few groups internationally are making really strong contributions. And I should mention there's sort of two paths to biomarkers that are both yielding a lot of new data and targets for drugs, which is sort of the serum or blood proteins that we've talked about, but also the genetic biomarkers. So the genetic modifiers, they're called, that are the differences in DNA between you, me, and everybody. But when some of those differences or polymorphisms that make us all individuals are superimposed on Duchenne children, they can have a dramatic effect. And it sort of makes sense. If somebody's a good athlete and their muscle is naturally strong, maybe that'll help a Duchenne kid be strong relative to other Duchenne children. So those both paths have really expanded since the early Wellstone days. To your question of the serum biomarkers, blood markers, so much of this is what's called context of use. So, a specific protein in the blood, you want to ask a specific question of it. It's any biomarker. I mean, plants opening their leaves to the sun is a biomarker. That's not really going to help us in Duchenne trials to see which proteins are responsible in a plant to open their leaves or their flowers. So, there is now more and more mature and informative context of use where specific biomarkers in Duchenne are used in specific instances and really greatly help a clinical trial or understanding of Duchenne or de-risking a drug to show that a drug is actually hitting its target and is worth a large clinical trial to get drug approval. I mean, to just give one example, through our biomarker work and large part through the Wellstone, we identified just 2 inflammatory proteins where they go up in the blood of anybody who's having like an inflammatory response, autoimmune or, arthritis or something like that. And we knew they were decreased by corticosteroids. So in many disorders, including Duchenne. So we could use those for vamorolone to show that depending on what dose of vamorolone we used, we were seeing a dose responsive or dose related reductions of those proteins. And that helped us greatly to understand, what dose of vamorolone we should be taking forward. And one of the nice things with the biomarkers is they can be acutely responsive, which means we measure those changes after just 2 weeks of treatment. And they very much predicted what happens 6 months later or a year later. And so that's an advantage of biomarkers. They can be predictive as well in certain contexts of use.
Yeah, Eric, I think Janet Woodcock has, in various presentations that she's given to our community, she has said inflammation plays such a huge role in this disease that if you could really control inflammation, you'd really have a different outcome. Obviously, we need other steroid or we need other ways to deliver the microdystrophin or to get it expressed through antisense and other ways. But she said, one of the fundamental issues is these kids are born, and as soon as they put their feet on the ground, this inflammatory component just goes to the sky. So can you tell us a little bit more about vamorolone? I know we complained to you. You were our sort of person to complain to, right? We first of all complained that no one was prescribing steroids when we had the belief that they were useful, right? So we were pestering you about that. And then as soon as we got them into sort of standard practice for most or many patients, at least to have the option to decide, then we came back and complained to you and said, except they're, they're hitting those adrenals so that boys aren't growing and they have a cushingoid appearance and they're gaining weight and the behaviors are out the door and their bone density is a mess. So could you please fix that? So as I say, I know we've used you as a little bit as our battering ram and come and said, here's a problem, can you fix it? So can you talk to me a little bit about where did you find valnorolone? What led you to finding it, because of all the side effects that we were seeing and really we find are so harmful to the quality of life of these young men and their families. So where did you find it? How did you find it? And what were your first steps?
Well, how we found it was actually directly through the Wellstone, in the sense that with the Wellstone grants, the government mandates that you have an external advisory board. So you must bring in external experts that review your progress. And one of our external advisors was John McCall, who was a chemist with Pfizer and Upjohn and Pharmacia, but had semi-retired and was doing a lot of consulting work, including for the National Institutes of Health. And so we brought him in as our advisory group for the Wellstone. And he said, years ago, I worked on this steroid-like thing, and it was used for brain trauma and spinal cord trauma. It was eventually dropped, but it's a membrane stabilizer, so maybe it'll help stabilize the dystrophin-deficient membranes. So it was really, that was the seed, the initial light bulb that Kanabwin Nagaraju and I and took into the labs and tried variants of his, what are called Delta 911 steroids, to see if they did stabilize membranes, which they did. But more importantly, that they separated out the effects of corticosteroids into efficacy and safety, where they were getting lots of hints that maybe we use this flavor of steroids, we'd keep the efficacy and start removing the side effects. So what's called a dissociative steroid, or it separates the efficacy from the safety concerns. And that has turned out to be true. In a double-blind trial with 121 kids we recently completed, we see the same benefit, quite marked benefit. And we did it in comparison to prednisone. Those kids get quite a bit better over 6 months, clear differentiation from placebo. On all the outcome measures we studied, 5 different motor outcomes, were all the same. And so it shows efficacy. Well, did it improve safety? Well, it did. We seem to have completely changed the bone biomarker, or the bone symptoms. So we see there's no longer stunting of growth. The kids actually grow a bit faster. It's like catch-up growth. And then the bone biomarkers, like we were talking about before, drops of blood, in drops of blood, you can see how bones are remodeling, and we've completely changed them opposite of what corticosteroids do. And it seems the mood disturbances also, you mentioned, Pat, the craziness that some kids on corticosteroids. We've had like a kid who got kicked out of nursery school because he was on corticosteroids, but going on vamorolone, he could go back to nursery school. And, because he was much better behaved. So yeah, I think we do seem to have removed, not all, but important side effects of corticosteroids. And it seems FDA says we have a good amount of data ahead towards drug approval.
So Eric, I'm sure you're publishing on that now that the study is completed. And when do you see a submission for a new drug application to the FDA? So that's the vehicle to get it approved in the US?
Sure. Usually the steps are that first you go to the FDA with what's called a pre-NDA meeting. So a new drug application is all the information you put together to give to FDA. They take about 6 months to a year to review it, and then they give you drug approval. So we had what's called a pre-NDA meeting, which is mandated, is typical, just last month. And they looked over our data, all the trial data, and said, yeah, thumbs up, you're good to submit an NDA. It doesn't mean we're automatically approved, but it says that their opinion is that the safety and efficacy data is sufficient for filing an NDA, which suggests that they look on it favorably as far as future drug approval. So we expect to send in the NDA early next year and hope to have approval later next year, early the following year.
That is pretty amazing. And so if we're thinking about gene therapy now, and these kids are, on a steroid regimen that's increased, do you foresee, or are you doing work around, or mouse work, or whatever work you have to figure out? Because having a drug that has less effect on behaviors, or no effect on behaviors, less effect on growth or none, and certainly doesn't impair bones or puberty. I'm guessing that you are not hitting that lever either. Do you see this then being substituted in the gene therapy studies, or how do you see this going forward?
Well, for both all the dystrophin replacement therapies, whether it's gene therapy, exon skipping, or CRISPR, they're all introducing not normal dystrophin, but abnormal dystrophin, sort of like Becker-like dystrophin. So they're highly modified dystrophins. And we know from all the patients that have similar types of dystrophin that they still have inflammation in their muscle and they still have muscular dystrophy. It's slowed down, it's like Becker. So all gene therapy trials and all exon skipping are done as an add-on to corticosteroids because you do still want to suppress the inflammation, as you mentioned, that Janet Woodcock mentioned is so central to the disease. So all dystrophin replacement efforts are a combination with corticosteroids. So we anticipate that once vamorolone is approved, then physicians and even clinical trials can have the option of either being on corticosteroids or on vamorolone with the add-on. The key thing is that it needs to be approved first. You can't combine investigational drugs because then you don't know what drug's doing what. But once a vamorolone is approved, then it becomes standard of care. And then the future clinical trials, say of gene therapy, would generally be encompass all forms of standard of care, which would be either corticosteroids or vamorolone. That's my guess. There is an additional use of corticosteroids specifically in gene therapy trials, and that's for induction of immune suppression. So I'm sure, as you know, Pat, that in the gene therapy trials, kids should be stably treated with corticosteroids coming in. But once they get gene therapy, they bump up the corticosteroids quite a bit to try to keep the body from rejecting the virus, so the gene therapy vector. So there you may envision a scenario where kids come in vamorolone and you do an add-on of corticosteroids and then wean off corticosteroids and stay on vamorolone. So there's a number of different possible paradigms. Towards that end, we've already switched over kids from corticosteroids to vamorolone. So in the trial we just finished, there were 30 kids that went from prednisone treatment to vamorolone treatment. And we just had a press release looking at that last week, and it seems like you saw the growth stunting with prednisone and gave them vamorolone, and they went right back up. So we could rescue at least some of the side effects of corticosteroids, which is promising. That it seems like switching between the two can be mostly good stuff instead of any additional safety concerns so far.
But I think the other important thing for families as well, if you're preserving the bone turnover, then you should theoretically have fewer fractures, because fractures, as you know, sometimes end ambulation, and fractures, as you know, can have more desperate consequences with fatty embolism. So I think having a drug that preserves growth and bone turnover should theoretically again limit the fracture risk. So it is an amazing opportunity. It feels like in the 20 or so years that we've known each other, and maybe even longer, 25, I'd say, things have progressed incredibly fast. Well, 25 years is not fast when you're looking at a child, and I recognize that as well. But it does, looking back, it's pretty amazing progress. If you had a crystal ball about advocacy, what would you want done, and what do you see as a future?
Well, as you said, Pat, we've come so far. When you and I started this, there were no clinical trials, no natural history studies, no biomarker studies. People didn't even know how to set up a clinical trial, much less run it. And now that's completely changed. I mean, there's dozens of clinical trials and all sorts of innovative ideas and different approaches. And I think, Duchenne is not an easy disease. I mean, it's in one of the largest genes out there. It's hard to deliver. Muscle is one of the largest target organs of the body. It's not like you can do a little gene therapy into one retina. You got to cover the whole body. So there's lots of hurdles, but there's lots of people working on it. And it's, to me, whenever you can set things up in parallel, so there's lots of things going on simultaneously, you're in a good spot. There's a good space, because something's going to hit, or any number of those things are going to hit. So it's no longer incremental one after the other, where you feel like it's just a shot on goal and the game's over. Now we have all these games going on in parallel with a lot of shots on goal. In terms of advocacy and what needs work, I mean, I think there's standard of care always has to be reevaluated, because things change. And so having that centralized and managed by you and the parent groups is so important, to make sure that all the kids and the physicians understand what is our current understanding of the best standard of care. So that's a key part of advocacy. Making data interchangeable between trials and between studies, so that people aren't reinventing the wheel, and that there's access not only between different scientists and researchers, but also data to the families participating in these trials. I mean, they should have access to their own clinical trial data, and right now that's problematic. And we're working on that, you're working on that, and I think you're doing a survey on that now, and that's so important. So there's lots of things that always can be made better, and the advocacy is so important to moving things forward.
Yeah, Eric, I totally agree with you. And I know as a parent, sometimes you don't think your voice counts, but clearly the parents, this community has really, and their advocacy has really changed the whole world of Duchenne much for the better, as we see going forward. We're incredibly grateful to you. As I say, I often think of you as our battery program. Fix this, do that, fix that. Can you help here? And I couldn't end this interview by not mentioning Joel Wood and his work with us together, as we all met and thought about how we could change things. We're really grateful to all that you've done. Can I ask, is there another drug that you have that you're thinking about after valmorelone, just to have some insight? Do you have other thoughts?
I think the key thing with exon skipping is to get higher dystrophin levels and seeing clinical efficacy. And so I think things are heading in the right direction there as well. And lots of different efforts to improve that. Lots of different efforts in gene therapy to try to reduce toxicities, see if you can get the best vector for the longest period of time. I think
ReveraGen, do you have more drugs that you're going to bring forward?
I don't know. I mean, probably there's other things we're thinking about as far as novel approaches to therapy. I mean, the key thing is to get full-length dystrophin into muscle. I mean, because Becker is always going to be Becker, and Becker is variable. And so you expect that some kids to gene therapy and exon skipping will react very well, and some not so well. And that's what we see in Becker muscular dystrophy, even with the same mutation. And again, it's this genetic modifiers and all these things that complicate an already complicated disease. So I think any effort to get full-length dystrophin, because that should be curative if you get enough of it into muscle. So yeah, we have some ideas for that, and others do as well. And so yeah, I think it's a very rich palette now of therapeutic approaches that didn't exist when we started. So it's been wonderful working with you, and I'm glad that you, I, and many people working together have made it quite a bit of the distance here.
Well, we're getting there. Eric, I can't thank you enough for spending time with us, but more than that, for Synergy, for Agata, for ReveraGen, for valmorelone, for listening to us, because you were the first one to allow us, if you will, into your lab to sit down and cry on your shoulder and say, how can we make this different for so many, and for our boys as well. So I can't thank you enough for all you do. And I hope to see Nova Scotia at some point. So
and I hope to have you up there. And please, I'm so grateful for everything you've done for all the families, Pat and Parent Project, and really holding the banner and keeping things moving and keeping everybody's eye on the ball. So thank you for all you've done.
We've got a great community. I think that what you and I can say is that we enable the community to come together with lots of foundations and lots of people and lots of ideas, and hopefully something we did worked right. But we're really anxious to see valmorelone come forward, and anxious to see what your next steps are. So even in Nova Scotia. So thank you so much, Eric.
Have a good day.
You too.
Bye.